Hi, everyone. My name is Matt Kaeberlein. I'm a professor of laboratory medicine and pathology at the University of Washington. I'm also the director of the Healthy Aging and Longevity Research Institute, and I'm the CEO of the American Aging Association. The title of this lecture is Targeting Biological Aging, a New Paradigm for 21st Century Medicine. This is really designed to be an introduction to the biology of aging. And what I hope to do is give you some insights into what we've learned about the mechanisms of aging. And then towards the end of the lecture, I'll present one intervention that seems to modify the biological aging process and has the potential to improve health and longevity for many people. So I want to start by talking a little bit about the way that we think about health and longevity, at least traditionally. And this is true both from the biomedical research perspective and also from the practice of clinical medicine. And what I want to point out is that traditionally we have really focused on individual diseases. And what I mean by that is that generally we think of somebody as healthy until they get sick, until they develop a disease, and then we try to do something about it. We either try to cure that disease or we try to at least alleviate the symptoms. And I really think of this as 19th century medicine or 20th century medicine, this focus on individual diseases. And one of the things that often gets lost by thinking about diseases in isolation by themselves is that all of these diseases shown here have at their root a single greatest risk factor. And it's not whether you smoke or how much you eat or how much alcohol you consume or whether you exercise, it's how old you are. So there's this underlying aging process that is responsible for enhancing the increased risk of developing all of these diseases. And by far, it's the greatest risk factor for these diseases. And so if we're able to understand what aging is from a biological mechanistic perspective, we have the potential to actually target those mechanisms to delay the onset and progression of all of these diseases at the same time. And so this is really what I think of as 21st century medicine. Where we're going is to really start to try to understand, what are the root causes of biological aging, and then use that information to slow the aging process, in some cases maybe even reverse functional declines that go along with aging to keep people healthier longer, by delaying the onset and progression of all of these diseases simultaneously. And there's been some effort put into trying to understand, what would that mean from the perspective of public health? And so now these are data that were derived from the CDC, the United States Center for Disease Control, which keeps very good statistics on what people die from. And so based on that, you can make an estimate. Well, what would the impact be for a typical 50-year-old woman if we could cure individual diseases? And this is work that I believe was first done by J.L. Shansky and colleagues to try to answer that question, to estimate what would the effect be on life expectancy. And so this sort of individual disease approach, 19th, 20th century medicine, even if we were successful, which we haven't been yet, even if we were successful at curing all forms of cancer, for a typical 50-year-old woman, that would only lead to about 3 years of increased life expectancy. The same thing is true if we were successful at curing all forms of heart disease. Again, really only about 3 to 4 additional years of life expectancy. And even if we could do both, if we could cure both of those, major killers, you still only get about 7 increased years of life expectancy for a typical 50-year-old woman. So, those effects, I think, are smaller than what most people would naturally assume, given how much attention and resources we've put towards curing individual diseases. At least at the population level, the impact on life expectancy is not so great. And I want to mention, I'm not trying to minimize the importance of curing an individual's disease. At the individual level, at the personal level, curing somebody's cancer is really important. But at the population level, not such a big effect. Now, if we compare that to the effect of slowing aging, by an extent that's pretty common now in laboratory animals at least, the effect on life expectancy, again, for a typical 50-year-old woman is proportionally much greater than targeting individual diseases. And that's simply because instead of affecting only one disease, when we target aging, we've pushed back all of these diseases of aging at the same time. the other really important point to make here is that those added years are mostly expected to be spent in relatively good health, free from chronic disease and disability. And that's represented by the green shading here. So really the prediction is that by targeting aging, we'll push those diseases of aging back later into life so that you get not only an increase in lifespan, but an increase in health span. And so this is a really important concept to understand. The goal here isn't so much about keeping people alive longer. It's about maximizing that period of life that's spent in good health, free from chronic disease and disability, and pushing those diseases of aging back as far as possible. Okay, so I have just a couple of major take-home messages that I hope you'll take away from this lecture. The first point that I want to make is that aging is just biology. And I make this point because I think for people in the general public or even scientists who don't study aging or think a lot about aging, when we start talking about slowing the aging process, it sounds sort of science fiction-y. And I just want to make the point, this is science fact, and that aging is just a biological process, much like development is. And this is pretty easy to see if you just look across the animal kingdom. So shown here are some animals that age at dramatically different rates, orders of magnitude. So there are fruit flies that in the laboratory will live a few months. A mouse in the laboratory will live a few years. We all know that dogs, depending on their body size, will live anywhere from about 8 to 10 years to, for small dogs, up to 20 years. And then there are some really interesting animals that live an exceptionally long time. So one example of this is this wrinkled pink guy here, the naked mole rat. The naked mole rat is genetically pretty closely related to other rodents like rats and mice, but it ages about 10 times more slowly. It lives about 30 years in captivity. And I'm not sure we really even know what the actual lifespan of the naked mole rat is, but it's certainly much longer than you would expect based on its genetic relationship to other rodents. So if we can understand what is it about the naked mole rat that causes it to age 10 times more slowly than its evolutionarily closely related animals, that potentially tells us something important about the biology of aging. And then there are animals that will live much longer than people do. There are whales that will live a couple hundred years. There are clams that will live 5 centuries. So again, nature has figured out how to modify the rate of aging by many orders of magnitude. And if you think about what's different about these animals, it really boils down to genetics. So of course their DNA is different, and environment. And that's just biology. So if we can understand what this biology is, then we have the potential to actually have an impact on the aging process and all of the degenerative declines that go along with that aging process. Which brings me to this sort of second take-home point, which is that we're starting to figure it out. And the field has actually made quite a bit of progress in understanding what the biology of aging is at a cellular, molecular, genetic, biochemical level. And so one way to think about this are these hallmarks of aging. So these are just evolutionarily conserved mechanisms that seem to be shared about the aging process, and many people in the field believe cause the degenerative processes that go along with aging. And so the hallmarks of aging are listed here, and over the next 10 minutes or so, I will just really briefly introduce you to each of these hallmarks of aging. But the point I want to make here is that aging is just biology. We've made a lot of progress at figuring out that biology. We can give names to these conserved molecular mechanisms of aging. And in principle, any or all of these hallmarks of aging are targets that can be modified to have an impact on the degenerative processes that go along with aging. Okay, so we're going to go through these hallmarks one by one, and as we go through them, I'm going to just give examples of how these hallmarks, even though we can categorize them as different things, really interact in a complex network to lead to the changes that we see go along with aging. So one of the hallmarks of aging is genomic instability. And this really just refers to changes to the DNA that happen during aging. And those changes can take many forms. So probably the form that most people would initially think of are mutations. And those can happen for a variety of reasons. They can happen due to damaging molecules like reactive oxygen species or ROS or radiation. They can also happen just as a byproduct of natural errors that occur during cell division, during replication. But these mutations certainly happen, we know that, and they accumulate with age. And so that's one form of genomic instability that accumulates with age. But what has been observed is that there are other types of genomic instability that also seem to happen during aging that might be as important, maybe even more important for some of the diseases and declines in function that we see go along with aging. One of those is activation of transposable elements. So these are DNA elements that exist within our genomes. Normally they're silent. They just sit there and don't really do anything. But with aging, we see that there is a loss of regulation of these transposable elements. They become activated, and that leads to DNA damage and genomic instability. And then another form of genome instability are terminally short telomeres, which we'll talk about as the next hallmark of aging. And the consequences of this genome instability are cancer. So we know that mutations are permissive and sometimes causal for cancer, can lead to cell death through a process called apoptosis, can also lead to cell senescence. And I'll talk more about senescence in just a minute. And one of the consequences of all of these things is an increase in chronic inflammatory signaling, or a process called inflammaging. And so this is one of the ways that genomic instability can have an effect on some of these other hallmarks of aging. The next hallmark of aging is telomere shortening, which I just alluded to. So telomeres are the caps at the end of our chromosomes, and they exist because during replication, we have a problem that it's not possible to replicate to the very end of the chromosomes through the normal replication process. So there are these repeated elements called telomeres that exist at the ends of chromosomes in cells that express an enzyme called telomerase. Those telomere caps are maintained, but most of the cells in the human body do not express telomerase, which means with every replication cycle, the telomeres get a little bit shorter and a little bit shorter and a little bit shorter. And what has been observed both in the laboratory, in vitro, in cells cultured in the laboratory, and in vivo in the human body is that at least in certain tissues, this telomere shortening process reaches a point where the telomeres become so short that they create a DNA damage signal. And that DNA damage signal then leads to either cell death or cell senescence. And that happens during aging and can lead to both the genomic instability that I just talked about and one of the other hallmarks of aging, which is cellular senescence. Epigenetic alterations are the third hallmark of aging. So epigenetic changes typically refer to chemical modifications of our genomes, of our DNA, or the proteins that surround the DNA, which are called histones. There are multiple types of chemical modifications, including methylation and acetylation and ubiquitination. And these chemical modifications can have significant effects on which genes are expressed, which genes get turned into mRNA and then get eventually translated into protein. And again, there's a large amount of accumulating evidence that these epigenetic changes happen during aging and contribute to many of the declines in function that go along with aging, and in particular, genome instability and cellular senescence. So again, links now between hallmarks of aging that are turning out to be important. The other thing that's worth mentioning about epigenetic changes during aging is there is a burgeoning field or subfield looking at these epigenetic changes as molecular clocks. And so it's been observed that these changes happen in a predictable way with age, and you can actually use the pattern of changes to create a clock, an epigenetic clock, which is highly predictive of chronological age. And shown down here in the figure are 3 different epigenetic clocks. And one of the things that a lot of people are interested in is asking, can these epigenetic clocks be used to tell us something about biological age, not just chronological age, how old is an individual in years and months, but biological age, how old are they in terms of their biology of aging. And so one idea would be that if the regression line here is a predictor of chronological age, so age is on the x-axis, and then predicted epigenetic age is on the y-axis, individuals who are far off of this line, either above the line or below the line, might be undergoing biological aging at a different rate. And so people who are above the line would be aging biologically more rapidly. In other words, their epigenetic age is greater than their chronological age, and vice versa. People who are below the line might be aging biologically more slowly. So that's still a little bit of a hypothesis, but there's accumulating evidence that indeed, at least to some extent, these epigenetic clocks can predict biological age and may actually play some sort of causal role. These epigenetic changes may actually play some sort of a causal role in some of the age-related changes that we see. The fourth hallmark of aging we're going to talk about is called loss of proteostasis. So proteostasis really just refers to the ability of our cells to maintain their proteins in the properly folded form. And of course, proteins have to be folded properly in order to function properly. And what has been observed is that with age, this proteostasis declines, and you get an accumulation of proteins that either misfold or become unfolded. They can become damaged, again, through things like reactive oxygen species or other chemical modifications. And when proteins become misfolded, they are often prone to aggregate. And we know that there are protein aggregates that accumulate during aging and are thought to be very important, at least in some diseases. So for example, protein aggregation during Alzheimer's disease has been for decades thought to be a part of the disease process that leads to neurodegeneration and neuron death. So one thing that's important to appreciate, though, is that when proteins lose their proper structure and they become misfolded, that can affect their function in a couple of ways. It can lead to a loss of their normal function, so they're no longer able to do what they need to be able to do. But they also can gain new functions, and this comes back to the idea of protein aggregation, that it's not only that you're losing the function of what the proteins normally do, they actually take on new toxic functions, which can then lead to problems like cell death or cell senescence. So again, there are clear connections between loss of proteostasis and some of the other hallmarks of aging. The fifth hallmark of aging is deregulated nutrient sensing. And this refers to the observation that nutrient sensing pathways seem to be among the most important, if not the most important, genetic pathways for regulating the rate of aging. And these include pathways like mTOR, which stands for the mechanistic target of rapamycin. I'll talk more about mTOR in a little bit. Insulin IGF-1 signaling, AMP kinase, sirtuins, all of these are well-characterized genetic modifiers of longevity that play a central role in sensing and responding to external nutrient availability. And it seems to be the case that it's the overactivation or hyperactivation of these pathways that's associated with faster aging. And the observation is, if you can turn down the signaling through these nutrient sensing pathways, that is associated with increased lifespan and slower aging in many different organisms. And I'll come back to this idea in a little bit, but this suggests the hypothesis that really the reason that nutrient sensing is so important during aging is that there's this balance between growth and reproduction on the one hand when nutrient levels are high, versus stress resistance and longevity when nutrient levels are low. And that these key sensing molecules, these nutrient sensing pathways, regulate this balance between fast aging and slow aging. And some of the consequences of deregulated nutrient sensing are increased cellular senescence, aberrant stem cell differentiation or stem cell senescence, and there are now connections between dysregulated metabolic signaling and the epigenetic changes that I just talked about, as well as loss of proteostasis. So again, it really seems to be the case that this hallmark of aging is interacting with other hallmarks of aging. Mitochondrial dysfunction is the sixth hallmark of aging. And to my mind, this is the one that I think you can really make a case, might be a root cause, because you can find evidence that mitochondrial dysfunction will result in all of the other hallmarks of aging, or at least it can contribute to them. So mitochondria are, of course, the powerhouses of the cell. They're where all of the ATP, which drives chemical reactions within the cell, they're where the ATP is generated. And one of the theories of aging that goes back many decades is that damage to mitochondria due to accumulation of reactive oxygen species, which are actually produced by the mitochondria, is sort of a foundational root cause of aging. This is related to the free radical theory of aging, or the mitochondrial free radical theory of aging. Now, the ROS that are produced as part of mitochondrial metabolism, as I've already alluded to, can lead to other forms of damage, like genomic instability, like damage to telomeres, like damage to proteins, which leads to protein misfolding and aggregation. So again, there's clear evidence that mitochondrial dysfunction can seed some of these other hallmarks of aging. We know that mitochondria become impaired in many tissues, maybe all tissues, but particularly in tissues where mitochondrial metabolism is really important, like heart and muscle and brain and fat. It's clear that mitochondrial damage with aging is contributing to declines in functions in those tissues. It's also the case that there are pathways that are evolved to help deal with this mitochondrial damage. So antioxidant enzymes that help alleviate the reactive oxygen species that mitochondria produce, or process called mitophagy, which is a specialized form of a general process called autophagy, where mitochondria can actually be recycled when they become damaged to create new mitochondria that don't have as much age-related damage. The seventh hallmark of aging is cellular senescence. And what this refers to is the observation that with age, there is an accumulation of cells that don't die, but they're no longer functioning the way that they're supposed to. So they are cells that have accumulated damage of some sort. And instead of undergoing cell death through apoptosis, they stick around. And so these cells are called senescent cells. And it's not so much the fact that they stick around and they're no longer doing their job that seems to be the problem. It's that when they stick around, they don't only stop doing what they're supposed to, but they start giving off chemical signals which are detrimental to the surrounding tissue. And these are largely pro-inflammatory signals, although not exclusively. And so this is called the SASP, or the senescence-associated secretory phenotype. And what has been observed is not only that with age, there's an increase in this chronic pro-inflammatory signaling, which is largely given off by senescent cells. It's also been seen that if you can reverse that, or if you can clear these senescent cells through a process called senolysis, that can actually lead to a reversal of the age-related inflammation or inflammaging, and increases in function and extension of lifespan. And so there's a lot of interest in developing drugs that can actually target and kill these senescent cells, which are referred to as senolytics. And again, as you might be recognizing, some hallmarks of aging can lead to cell senescence or contribute to cell senescence, and then cell senescence itself can have effects on other hallmarks of aging, like stem cell differentiation and intracellular communication through this inflammaging process. The next hallmark of aging is stem cell exhaustion, which I just alluded to how stem cell senescence can affect this. This just refers to the fact that in most tissues, maybe all tissues, during aging, there is a decline in stem cell function. It's a little bit unclear if the actual number of stem cells declines, but certainly the function of those stem cells declines along with the aging process. And this can obviously lead to tissue and organ dysfunction, and can contribute to many of the phenotypes that we see that go along with aging. This stem cell exhaustion is due both to intrinsic factors, and what I mean by that are changes within the stem cells themselves, as well as extrinsic factors, so changes in the niche that the stem cells exist in. And it's the combination of both these intrinsic and extrinsic factors that lead to the stem cell exhaustion. I've already mentioned the important role that senescence probably plays in stem cell exhaustion, and that this stem cell exhaustion can lead to many of the important diseases that we see with aging. So for example, obvious diseases that likely arise from stem cell exhaustion are age-related cancers. So the immune system in particular is critical for surveilling and detecting cancers early and then removing them. As we get older, there's a decline in immune stem cell function, which leads to an increased risk of age-related cancers. Probably also contributes to age-related dysregulation of the microbiome, and then also contributes to the increased risk of infection in the elderly. And I'll come back at the end of my talk and talk a little bit about this in the context of COVID-19. So stem cell senescence, one obvious example where this is critically important to age-related disease is senescence of the immune system. And we now know that there are some interventions that can actually restore stem cell function in aged animals. Senolytics, I've already talked about. Clearing senescent cells. Rapamycin is a drug that I'll talk about more in a few minutes. And then there's this experimental paradigm called parabiosis, or heterochronic parabiosis, where the circulatory system of a young animal is surgically connected to the circulatory system of an old animal, so they share blood. And it's been observed that this heterochronic parabiosis can actually restore stem cell function in the old animal, at least in certain tissues. So that leads to the conclusion then that there's some circulating factor or multiple circulating factors that are driving this stem cell senescence. And I think, inflammatory factors are probably one of the circulatory components that play a role in this rejuvenating effect on stem cells of heterochronic parabiosis. And so then the last hallmark of aging that we're going to talk about today is altered intercellular communication. And so really this just refers to the fact that in order for a complicated multicellular animal to function properly, the cells have to communicate with each other. And they do that through a variety of mechanisms. So there are hormones that are given off and circulate systemically throughout the entire body to regulate the function of other cells. There are metabolites that are secreted by some cells and taken up by other cells. There are chemokines and cytokines or signaling molecules. All of these are critical ways that cells within our body communicate with their neighbors, maybe right next door within the same tissue, or with other tissues that are in distant parts of the body. And again, what has been observed is that during aging, all of these forms of intercellular communication become perturbed. They don't work the way that they're supposed to anymore. That's seen at the level of hormonal changes. Growth hormones, sex hormones are commonly recognized to change with aging and contribute to age-related phenotypes. Muscle loss in the case of growth hormone with aging, menopause in the case of sex hormone changes. But that happens with lots of other hormones as well. So there are many hormonal changes that occur during aging that lead to changes in our physiology and some of the phenotypes that we see with aging. There are also niche changes I've already alluded to that contribute to stem cell exhaustion, and inflammaging, which I've mentioned several times, which is driven at least in part by cellular senescence. And then what is probably particularly important is metabolic deregulation that we see go along with aging. And that comes back to this just dysregulation of the nutrient sensing pathways. So these are all examples of altered intercellular communication having an impact on the biological aging process. Okay, so those are the hallmarks of aging. And now what I want to do is shift gears a little bit and talk for a minute about some of the approaches that scientists take to study aging. And there are lots of ways that this can be done. And I just want to briefly mention a few of them. So one approach that has been taken to study the biology of aging is to study people who appear to age more rapidly than the average person. And so I think we all recognize that if we just look across even people that we know, there are people who appear to be aging more quickly than other people. But then there are rare diseases where individuals undergo what looks like accelerated aging at an even much more rapid pace. So one example of this is a disease called Werner syndrome, where people in young adulthood will start to show these symptoms of accelerated aging. And by the time they're in their 40s and 50s, they look like somebody who's in their 70s, 80s, 90s. And the genetic basis for Werner syndrome is known. And one approach that has been taken in the field is to try to understand, well, what are the mechanisms that lead to this apparent accelerated aging? And even more severe disease is a disease called Hutchinson-Gilford-Progeria syndrome, which is caused by a mutation in the gene lamin A. And this leads to accelerated aging phenotypes on the order of 15 years. I think one of the things that's important to appreciate is that all of these accelerated aging disorders are really what are called segmental progerias. And this refers to the fact that they don't seem to have accelerated aging in every tissue. It's a subset of tissues that are really driving the apparent accelerated aging rate. And so they're not a perfect model of every aspect of accelerated aging. Sort of the reverse approach from studying people who appear to age rapidly is studying people who appear to age slowly. And so these would be people who live an exceptionally long period of time and or maintain health until very late in old age. And so this is a picture of the longest documented lived human being, Jean Calment, who lived to 122 years and 164 days. Jean Calment is a very sort of interesting person in the sense that, she was clearly active late into life, but she didn't really practice all of the healthy lifestyle practices that we might attribute with expected longevity. So she quit smoking at the ripe age of 117. And she ascribed her longevity to olive oil, port wine, and chocolate. So I think this is an interesting example. Obviously, it's anecdotal, but this is seen sometimes that the people who live exceptionally long aren't always practicing healthy lifestyles. And so this has led to the idea that maybe the exceptional agers have a genetic background that makes them resistant to some of the insults that would normally lead to disease and death in the average person. That's still a hypothesis at this point, but there's some evidence for that. And it's clear that there is a genetic component to human longevity. Right now, the estimates are that about 25% of human longevity is heritable, and it is absolutely the case that siblings of people who live to be more than 100 years old, so centenarians, are themselves also more likely to live to be more than 100 years old. And their offspring are more likely to live to be more than 90 years old. And so there are people who study the genetics of these families to try to understand, are there specific genetic variants that are enriched in these people who live an exceptionally long time? Another approach is what are called longitudinal studies of aging. And so this really just refers to following individuals over their lives and studying them to try to understand what are the most important genetic and environmental factors that influence different trajectories of aging in different individuals. And so these are extremely powerful approaches, and there are several longitudinal studies of aging that have identified genetic variants that are potentially important for human health and longevity, and environmental components that are potentially important for human health and longevity. There are also some limitations to these kinds of longitudinal studies. So one is that sort of by definition, these observational longitudinal studies will only identify correlations. They will identify things that are associated with health or disease, but they can't identify causation. And so they're hypothesis generating, but it requires then additional studies to go back and test those hypotheses to determine causality. They're extremely expensive and time consuming. So, to follow a typical human being from birth to death, we're talking 9 decades. So that's generally not feasible from the perspective of scientific studies, although there are some of these longitudinal studies of aging that have been going on for multiple decades so that they can capture, at least a significant portion of the adult lifespan for humans. One of the challenges, though, with that sort of, long time frame for these studies is the technology becomes outdated. So the same technologies that were used, 30 years ago, some of them are the same. Some of the clinical tests are the same, but many of the technologies have have progressed. And so we don't always have the same type of data for people who were studied 30 years ago as we will, even for the same people today who can be studied with some of these new technologies. Another approach to understanding aging is comparative biology of different animals, and in particular, these exceptional agers that I alluded to earlier, like the naked mole rat. So these animals that live longer than we might expect them to, or much longer than humans. By studying them, if we can understand something about their biology and what makes them age at a much slower rate, that potentially will give us insights into mechanisms of aging that are important for humans. And then I would say what has had the greatest impact on the field over the last 20, 25 years are studies that have been performed in the laboratory in model organisms like the budding yeast, Saccharomyces cerevisiae, nematode C. elegans, the fruit fly Drosophila melanogaster, which is not pictured here, and mice, to try to understand what are some of the mechanisms of aging that are conserved across these different organisms, with the prediction that if particular mechanisms of aging are shared in yeast and worms and mice, there's at least a reasonable chance that those mechanisms will also be shared in humans. Now, the power of these model organisms are, first of all, they age much more rapidly than people do. So on the order of a few weeks for yeast and worms, a few months for flies, a few years for mice. And so that means that scientists can do studies in a time frame that's sort of amenable to real mechanistic understanding instead of just correlation. And so that has allowed the field of aging research to identify these genes and pathways that, at least in these model organisms, clearly play a fundamental role in the aging process. It has also been what has allowed the field to identify the hallmarks of aging that I talked about earlier. And so one of the outcomes of those kinds of studies has been the identification of both genetic pathways that play a conserved role in regulating the aging process across all of these different organisms, and interventions that target these pathways and target the hallmarks of aging to have an effect on lifespan and health span in these different model organisms. And so this slide just shows some of the genetic pathways and some of the interventions, which I've grouped under lifestyle interventions or pharmacological interventions, drugs. All of these things listed here have been shown to affect lifespan in these different model organisms, and they're studied still fairly extensively in the field to try to understand what exactly are the mechanisms by which these different interventions are working. Now, one of the things this has led to is this appreciation that most of the interventions that have been studied so far are targeting these conserved nutrient responsive pathways. And so now I want to come back to this idea of, why is it that aging is conserved? You might not expect that the biology of aging would be the same in a yeast and a nematode and a fruit fly and a mouse and potentially in people. And so I think we can make an evolutionary argument for why aging might be conserved. And it comes down to the idea that there's this fundamental decision that all organisms have had to make from an evolutionary perspective. And that is to sense the environment and make the decision, given the environment that I'm in now, from a selective perspective, is it advantageous to grow and reproduce? Or is it better to shut down growth and reproduction and become stress resistant? And the idea there is that by shutting down growth and reproduction and becoming stress resistant, if it's a bad environment, you might be able to survive until the environment returns to a condition that's favorable for growth and reproduction. So if you think about what are the cues that, again, looking back through evolutionary time, all organisms have had to look at in the environment, one of the most important is nutrient availability. So is there enough food that it makes sense to have babies so you'll be able to feed your babies? And if not, then it's probably not a good time to grow and reproduce. And so these nutrient sensing pathways have evolved, and they've maintained that function over an evolutionary timescale of sensing nutrients and allowing the organism to appropriately make the decision between growth and reproduction versus stress resistance and longevity. So this may explain why pathways that regulate aging are shared across all of these very different organisms. One point that I think it's important to appreciate here is that under this model, natural selection did not act on the role of these pathways in regulating aging. That's sort of an accidental byproduct. What's really being acted on here is the role of these pathways in regulating growth and reproduction or stress resistance. It just turns out that by shutting down growth and reproduction and by turning up stress resistance, that has an effect on the rate of aging. And that makes sense, but it's really important to appreciate that aging itself, we don't think, is under evolutionary pressure or under natural selection. It's these other functions of growth and reproduction and stress resistance where natural selection is acting. And that also fits with the observation that these genes and interventions that have been discovered, often through unbiased approaches, seem to predominantly affect nutrient signaling pathways. And so what I want to do now is just finish up by talking about one of these specific nutrient sensing pathways, the mechanistic target of rapamycin, or mTOR, and the drug rapamycin, which is a specific inhibitor of this pathway. And I'm gonna focus a little bit on mTOR and rapamycin for two reasons. First of all, it is sort of the gold standard in the aging field for a pathway that affects aging. So it affects aging in all of these model organisms, and it appears to be robust across many different labs, many different genetic backgrounds, and the magnitude of effect that is seen reproducibly is larger than most of these other interventions. So it seems to be one of the most robust ways to modify the rate of aging across evolutionarily divergent organisms. So I mentioned that mTOR is a nutrient sensing pathway, and it actually senses nutrients through a couple of different mechanisms. So mTOR directly can sense the amount of protein that's taken in through dietary methods. So it can directly sense amino acids, particularly branched chain amino acids, and leucine will bind and signal through mTORC1. It's also sensitive to these hormonal factors like growth hormone, insulin signaling, IGF-1 signaling. Those also can act to regulate mTOR activity. And it's also appears to sense stress signals. And so there's still a lot of work going on in exactly how mTOR is sensing all of these different signals. But it's clear that it is responsive largely to different types of nutrient signals and also to different types of stress signals. And it's primarily one of two mTOR protein complexes that we think is most important for aging, and that's mTORC1, or mTOR complex 1. And this is the complex that rapamycin inhibits. So I said rapamycin is a drug that inhibits mTOR, and particularly mTOR complex 1. And mTOR complex 1 has an effect on multiple different processes that are likely to be important for aging, including stem cell turnover, cellular senescence, inflammation, protein synthesis, mitochondrial function, and autophagy. And I think it's still being worked out exactly how mTOR is affecting all of these downstream processes. One of the things that's important to appreciate, though, is that it's unlikely that just affecting one of these things is going to give you the same benefits in terms of effects on aging and lifespan as something like rapamycin. That's a prediction that hasn't definitively been shown, but I think the fact that mTOR regulates all of these different downstream processes, which are likely important for its effects on aging, makes it unlikely that we're going to be able to just target one of these and get the same effects as rapamycin. So what is rapamycin? Rapamycin is a drug that, as I mentioned, and it's got sort of a really interesting backstory. So it was actually first identified on Easter Island, which is also called Rapa Nui. That's where these statues come from. And that's where the drug gets its name from. As I mentioned, it's a specific inhibitor of mTOR complex 1. It's the most effective longevity drug to date. In mice, it increases lifespan up to 30%. There are a couple of things that are important to appreciate about rapamycin. The first is that you don't have to start giving it, at least to mice, from a young age. You can start the treatment in middle age and get essentially the same effects on lifespan. And it also is important to appreciate that it's not just affecting lifespan. It seems to broadly delay, and in some cases even reverse, the declines that go along with aging in many different tissues, including brain, heart, immune, muscle, oral cavity, kidney. So it really seems to be the case that rapamycin is affecting the intrinsic biological aging process and extending health span at the same time that it's impacting lifespan. This is just the data showing that if you start the treatment in middle age, you get about the same effect on lifespan as starting at young age. So this is work from the National Institute on Aging Interventions Testing Program. The graph on the right shows the effect of starting treatment with rapamycin at 9 months of age. The red are the mice that got rapamycin. The blue are the mice that got the control. You can see the red mice have increased median and maximum lifespan. And if you start the same treatment at 20 months of age, which is about maybe the mouse equivalent of a 60-year-old person, you still get about the same effect on median lifespan and maximum lifespan. So a midlife treatment with rapamycin gives almost the same benefits in terms of lifespan as lifelong treatment. Turns out that transient treatment with rapamycin can also give quite robust benefits in terms of lifespan. So this is work published from my lab a few years ago where we treated mice starting at 20 months of age, again, about 60-year-old equivalent of a mouse. We treated them for 12 weeks with either rapamycin or the control, and then we stopped the treatment. And we just put the mice back on the shelf in their cages and let them live out the rest of their lives, and asked, what was the effect of the treatment? And it turns out that if you look at survival after the end of the treatment, there's about a 60% increase in life expectancy in this particular cohort. And a couple of interesting things to note about that. The actual length of the lifespan extension in this case was 141 days, but the length of treatment was only 90 days. So the increase in lifespan actually exceeded the length of time the mice saw the drug. And obviously we don't know if this is going to extrapolate linearly to people, but if it did, that would be about 2 decades for that same 50-year-old woman that I mentioned towards the beginning of my talk. So again, these are potentially pretty significant effects on lifespan from a transient treatment with rapamycin in middle age. Now, why is it important that midlife treatment or transient treatments can have effects? This is really important from a translational perspective. If you think about an intervention that you might actually move into the clinic to have an impact on aging in people, you don't want something that teenagers have to start taking. You want an intervention that can be administered to people in middle age when they're starting to see these declines in function that go along with the aging process. And rapamycin appears to be such an intervention. What I think has been particularly exciting over the last few years has been the observation that not only can transient rapamycin have effects on lifespan, but it actually reverses many of the functional declines that go along with aging in very different tissues. So there's data in both mice and companion dogs that short-term treatment with rapamycin can reverse cardiac aging. There's data in mice that short-term treatment with rapamycin can reverse periodontal disease. And there's data in both mice and a little bit in people that short-term treatment with rapamycin can reverse immune decline. And I'm going to just spend the next couple of minutes talking in a little bit more detail about these effects on immunity, because I think this is really important for a potential example of how an aging intervention might make it into the clinic. So this is an experiment from Pan Zheng's lab at the University of Michigan, where they asked, what is the effect of 6 weeks of treatment of rapamycin on the aged immune system in mice? And so the way this experiment was designed is there were either young mice or old mice. In this case, the mice were 24 months of age, which now we're talking, maybe 65, 70-year-old mice. They were treated for 6 weeks with either control or rapamycin. The control is called the vehicle, that's this VEH. 6 weeks of either rapamycin or the vehicle. And then there was a 2-week washout period before they were immunized with a flu vaccine. So this is the H1N1 strain of influenza. They got a flu vaccine, and then they waited another 2 weeks, and then they were challenged with influenza to see, did the vaccine work and are the mice protected from this influenza challenge? So this is the survival outcome of that experiment. So the first thing to look at is what happens in a young mouse. If you're a young mouse and you don't get immunized, that's the solid line here. No immunization, challenged with influenza. This is a lethal dose of influenza. All of the mice died within 8 days. Okay, that's what you would expect. No vaccination. If you're a young mouse that gets vaccinated, you're completely protected. Again, this is what we would expect. vaccination, young immune system, the immune system functions the way it's supposed to. The mice are protected when they're challenged with influenza because they've been vaccinated. So those are sort of the controls. Now, what happens if you're an old mouse? This is where it gets really interesting. If you're an old mouse and you did not get rapamycin, you only got the vehicle, you get the immunization, but there's a 70% chance that your immune system does not respond to that immunization. And when challenged with influenza, you die, okay? This is the aging process right here in black and white. The aged immune system does not function the way the young immune system functions. And this can be seen in the case of a flu vaccine. This is also true in elderly people. They don't respond to a flu vaccine as well as young people. If you're an old mouse and you get 6 weeks of rapamycin, there's a dotted line here, right under the young immunized mice. The immunization is 100% effective. You're completely protected against influenza challenge. So at least for this measure of immune function, response to a flu vaccine, rapamycin took the aged immune system and made it function like that of a young mouse. So rapamycin reversed immune aging, at least as measured by response to a flu vaccine. There's pretty good evidence that the same thing is probably true in people. There have been 2 phase 2 clinical trials with a derivative of rapamycin called everolimus. It works exactly the same way as rapamycin, just has a little bit different bioavailability. Both of those studies showed that healthy older people treated with 6 weeks of everolimus had improved flu vaccine response. And then the second study showed that these healthy older people were actually also protected against other types of respiratory tract infections over the next year. So again, really seems to be the case that inhibition of mTORC1 with rapamycin can rejuvenate immune function both in old mice and also in old people, which is really exciting, which suggests that at least this role of mTOR in the aging process is probably shared in humans. Okay, so with that, I'll just wrap up with the summary. What I've told you today is that the biology of aging is being solved. We have information now about the hallmarks of aging, which are conserved mechanisms of aging. Targeting aging is likely to be more effective than disease-specific approaches, and that's 21st century medicine. Each of the hallmarks of aging represent targets for intervention to delay or reverse aspects of biological aging. And we already have sort of first generation longevity interventions, and I spent some time talking about rapamycin. And rapamycin, I believe, is a leading candidate for increasing lifespan and health span in people with few side effects.